So dear students and friends, okay, this lecture I'm going to discuss about the secondary metabolite production. Okay, so what is mean by secondary metabolite? Okay, first and foremost, you should know the difference between primary metabolite and secondary metabolite. Okay, so metabolic process, that is the synthesizing of different components in plants. Okay, you should know the first, what is meant by primary metabolite. Yes, it's a larger quantity. What are the primary metabolites? Proteins, carbohydrate, fat, and oils. They will synthesize in larger quantities in plants. Okay, that is for helpful for our food and other purpose. Okay, the carbohydrate, protein, fat, and oils, they will synthesize in larger quantity in all kind of plant species. That is primary metabolites. They will useful for growth and development of your plants. Similarly, the secondary metabolites. Secondary metabolites, it is synthesizing in plants very meager amount, very little amount, very little amount they will synthesize. What is the use for this? Okay, if you take example, the classical example in turmeric, in day-to-day life we are using, and also the neem. Yes, the neem leaf, neem bark, and other parts we are using like insecticidal properties. They having acidic acting. In curcumin, in turmeric, they will synthesize in very less. So what is the use for this? So if you take in aloe vera, if you take in allium cepa, if you take different kind of medicinal plants, so sweet and bitter and sour, and even they having different kind of properties. That is, they will synthesize in meager amount, very less amount they will synthesize, but they will using for our medicinal purpose. Okay, why they are synthesizing in the plants? That you should know the answer. Okay, so anti-cancer properties in turmeric, anti-azadirachtin, they having the insect control, that is the bioinsecticide. Okay, why they are synthesizing in the plants? We'll see this. So the plant secondary metabolites, the main function. Okay, the primary function, they're having defense and attraction. Okay, so mainly they are synthesizing very less amount, but what is the defense? Okay, for externally in plants having thorns and thick cuticular layer and bark very hard in nature, they will protect from pest and disease, even they will protect from abiotic stress. But inside the plants they will synthesizing in the meager amount. For example, herbivores, insect and vertebrates, okay, they will feed on the plants. While what will happen while feeding in the plants, they will segregate some things, that is for toxicity or growth inhibition or deterrence or repellents. Okay, if you feed on the plants, they will segregate this hormone. And also if you feed on the bacteria and fungal and virus, they will produce some toxic material to protect from the microbes. Even competing plants, you know the allelopathic effect, whilst root exudates, that root exudates never allowed to grow other crops. Okay, they will inhibit the germination, or they will inhibit the root growth. That's why if you see in the roadside, in the tamarind seed, tamarind plants, tamarind trees, down the tamarind trees, never growing other plants. What will happen? They will segregate the secondary metabolites. They won't allow other plants. Okay, first they will protect from insects and pests and protect from microbes, even protect from competing from other plants. So they will giving defense mechanism. They will segregate very less amount. Okay, this is the main function for the secondary metabolites. And also the attraction. So attraction, so pollinating insects, so smells, they will segregate some smells to attract the insects. And seed dispersing animals, yes, and root nodules bacteria, adoption of herbivores. Okay, because this is the things. So flower, flower nectar and smells, different kind of flowers having different kind of smells. That is the secondary metabolites. Okay, that's not a primary metabolites. Primary metabolites, carbohydrate, fat, protein, and oils. But secondary metabolites, they will synthesize very less amount. For example, classical example, jasmine. If you take the leaf, never going to smell is there, jasmine smell. But if you take the flower having this smell, so the jasmine now they are doing for extraction of scent, that is for the spraying, body spraying, and fragrance they are extracting. Okay, and also if you take the chilies, if you take the leaf of the chili, so never going to they're having the flavors, and also most of the spices they're having the flavors, condiments, they will synthesize very less. And also for the UV protection, so the function is for protecting from light and nitrogen storage. Okay, so what are the secondary metabolites? Terpenoids, polyphenol components, nitrogenous components. So they will activate the enzyme activity. Okay, different kind of enzyme activities. If you feed on the insect, or if you feed on the microbes, or even feeding of the other injuries, they will redirect this primary metabolite to secondary metabolites. Okay, at the signal going to prime, the signal transduction, they will to, yes, the insects going to attack. Okay, so the terpenoids going to act, phenolic components and nitrogenous components, they will alkaloids, glycolides, glycanoids, and okay, so polyphenols and terpenoids, they will activate and to protect the microbes and protect the insects, but they will segregate in very less amount. Okay, you should know the difference between secondary metabolites and primary metabolites. Okay, so if you take the classical example, so antimicrobial drug, insecticide and painkiller, medicine, snake bite and central nervous system stimulant, antiseptic and sedative and insecticides. So the classical example, nicotine. So nicotine having nicotina tobacco. So the leaves having the nicotine content, they will use as a bioinsecticide. Okay, and quinone. Okay, the Cinchona officinalis. Okay, bark. They having the antimicrobial drug, and morphine and capsaicin. Okay, the papivorum somniferum. They having the fruit having painkiller properties. Okay, caffeine. Coffee. The seeds having the caffeine content. Okay, so central nervous system stimulant. Okay, and also nimbin or azadirachtin. They having azadirachta indica, neem, the seed, box, and leaves having antiseptic and insecticidal properties. And the chrysanthemum flower, they're having pyrethroids. Okay, the flower having insecticide properties. Okay, so the pyrethroids using for killing of mosquitoes, so that the plants pots having the properties the alkaloids having. Okay, they will ask the question nicotine and okay pyrethroids. For the point 5, they will ask what is the secondary metabolites, which alkaloid present in the chrysanthemum, which alkaloids present in the tobacco plants. Okay, which part they having, which part of the flower which part of the plants having pyrethroids, and which part of the nicotina having the part having nicotine. Okay, the morphine, they will ask painkiller. Okay, papivorum somniferum. Okay, opium plants, they having painkiller. These are the alkaloids. These are the classical example for secondary metabolites. Okay, so anti-diabetic properties and dyes. So they will ask synconin. Okay, synconin dye, the landospermum erythrorhizine. They will ask that dye. Okay, which dye is, and also antidiabetic stevia plant. Garlic and Gymnema stylorus. Gymnema stylorus is they will call us in Tamil, they will call us chakra kolli. They have gymnic acid, the properties for to regulate the insulin. Okay, and also to reduce this, and also fenugreek. The fenugreek, you have triquinoline and nicotinic acid and coumarin. Okay, they having the properties to stabilize the glucose level. Okay, fenugreek having the alkaloids, triquinoline and nicotinic acid and coumarin. The three things are present in vendaethala. Okay, they having stabilized the glucose. These are really meager amount. That is anti-diabetic properties. And also if you take marigold, okay, safflower and betalin and beta cyanin, okay, beta cyacin, the beta vulgaris. Okay, this beta vulgaris, that is for this one sweet potato, they having the dyes. Okay, and corcosine and corconoids, the corcus sativus, these are the properties they having the isolation of dyes. These are also very less amount they are segregating in the plant pots. And also, essential oils and flavors and fragrance and pharmaceutical components. Take the different kind of essential oil, rosmarinic acid, the rosemary plants, they're having rosmarinic, Asimum bacillum, okay, Asimum bacillum, they're having rosmarinic acid, that is for this salva fructosa, and also menthol, menthol, mentha avaris, okay, then sweet orange having volatile components, okay, linalool and sinoil, thymus mesticana plants, okay, vitis vinifera, that is monoterpenes, okay, vitis vinifera, they having the essential oils, okay, peppermint having cornovine and menthanol, okay, so different kind of plants, the vetiver, they are also having the essential oils they are using for the fragrant agency, and also the essential oils also secreting in plants very meager amount, and also the flavor and fragrance, the caffeic acid, okay, capsicum fructose, and saffronella, okay, Quercus sativus, and vanilla, vanilla flavor ice creams we are taking now, vanillin planifolia, the vanillin content is present in vanilla beans, okay, so this is one of the commercial important fragrance. They will ask the question, vanilla planifloria, which part of the plants they having vanillin properties? The beans of the vanilla, okay, so one of the commercial important, okay, and also the pharmaceutical components they will ask anti-cancer principle, okay, they will ask this important question, taxus brevifolia, oh, this is very very important question, Taxol, okay, Taxol, they are isolating from Taxus brevifolia. This is one of the tree having the anti-cancer properties. The first isolated secondary metabolites. Okay, so now we seen about the alkaloids. What are the alkaloids? What are the parts of the plants? Then so dyes, antidiabetics, and essential oil, flavors and fragrance, and pharmaceutical components. Okay, and Cantharellus roseus in the cantharin. Okay, Cantharellus roseus, it is blindly available in our college campus. Everywhere you will see this Cantharellus roseus. They having cantharin, they having anti-cancer properties, and also if you take this, say ginsenoid, okay, Panax ginseng plant also having this pharmaceutical components. Now everybody is talking about the coronavirus having the, so any plant-based medicine is possible. So now they are government also working with this for isolating the components. They will use antiviral properties. The ginseng having the antiviral properties. Okay, so now they are using different kind of plant parts, different things. Their research is going on. Okay. So how we are isolating the secondary metabolites, which method is formulating? So already we talk about suspension culture. We isolate, take the plants, which plant we culture it, and cells we isolate, and mass multiplication of the cell, then we isolate, and solvent use and extract with the help of downstream processing. We have two novel components we will isolate. So how they are comparing? Okay, this is the cell suspension culture. They are going to use secondary metabolites. Okay, so that is incubator shaker. We are mass multiplying the cells. The targeted tissue, we are isolating the secondary metabolites. Okay, through cell culture. That is cell suspension culture we are using. And also bioreactors by using of hairy root culture and cell and hairy root culture. So already I told agrobacterium rhizogenesis, the bacteria will infect the plant parts. They will develop the roots. They're having the plasmid, RI plasmid, that is root induced plasmid, so that the roots will be formed out. And also bioreactor, it's a controlled condition nutrition. We are culturing the plants cells. We can mass multiplying the cell, extracting. So this is the big size bioreactor for industrial type. So isolating the cells and isolate the cells and grown up, and how much kg you want, we can grown up the bioreactor, isolate the cells and extract the novel components. This is the secondary metabolic application point of view. Okay, and also I told already biopigment and dye. So culturing the part of the plants and take the callus induction. The callus induction we can use and culturing it and isolate the components. This is biopigments and dye. Okay, so how to improve the yield is a very 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 important. I told already very less amount only synthesis in the plant parts, but we need to synthesis more amount. How? Addition of precursors. Okay, so if you're adding, so we should know the pathways. So okay, which pathway which the component is developing. So we need to make precursors, adding of precursors to increase the yield of metabolic yield. Okay, they will ask what are the methods to improve the secondary metabolic yield, and biotransformation to identify the genes, the novel components genes will transfer the plants and extract larger amount. That is a biotransformation. So that is called molecular farming. Molecular farming. Take the genes, for example, azadirachtin indica. Azadirachtin, which gene is responsible to form the insecticidal properties. Take out the genes and transfer it to the tobacco plants or other cultivable plants. We can extract. And elicitor treatment. Elicitor means, for example, some of the microbes will feed on the plants, then only they will develop the components. If you in the fungal infection take the bark, then only the bark they will segregate these components, and also the smell is coming. Okay, cinnamon bark, we are using our biryani. That is the cinnamon bark. Some the fungal things, the fungi they will feed on the bark, then only they will fragrance will come out. Without infection of the fungus, they never going to the flavor will come out. Okay, so the elucidate to elucidate the components with the help of even we can chemical elucidation, adding up some chemicals to elucidate to develop the secondary metabolites, and also mutagenesis. Yes, by using of chemical mutation, so okay, the mutation will helpful for to synthesizing amount. Okay, the so addition of precursors, biotransformation, and elicitor treatment, and mutagenesis. Okay, these are the yield improvement. Okay, how they analyzing the secondary metabolites? They will ask which instrument they will use for secondary metabolite yield and quantification. That is high performance liquid chromatography. High performance liquid chromatography, that is called as HPLC. HPLC, the role. So if you take the components, for example, azadirachtin, we should know the chemical component. So we need to compare. Okay, we need to compare. For example, take the leaf, bark, leaf seeds, and leaf pods. In even the embryo of the plants are matured seeds. We are taking all the part of the plants, which part having more amount of azadirachtin. Okay, which part having the azadirachtin, we should compare the peakness. Okay, to compare similar like all kind of plants. Okay, we should know the components, which part having the components. Okay, we can isolate easily, even take the tissue and culture it and isolate the things. This is the analysis part and quantification part, how much percentage. So we need to control. Okay, always we need to control. This is synthetically available, which components and the chemical formulas we should know. So from this control we should compare the which part of the plants we are going to extract. Okay, so up to this we talk about the secondary metabolites, and what is meant by secondary metabolites, primary metabolite and secondary metabolites, what are the components. Okay, how what are the uses and functions of the secondary metabolites, how they are synthesizing, and what are the parts of the plants, what are the plants having the, what are the uses we talk about, and antibiotics, diabetics, dyes, and flavors and fragrance, essential oils, and how they are culturing and isolating, and how they are larger cultivation of the secondary metabolites and biopigments. Finally, what are the methods to improve the yield of secondary metabolites, and also analysis part and quantification. Up to this you should know about the secondary metabolites and the uses and quantification. Okay, thank you.